Okay. As Professor Cavalli has mentioned, and it was always a pleasant surprise for a researcher, basic research is taking more and more of a role in meetings that used to be much more clinical. And in fact, the results of basic research are getting closer and closer to clinical translation and application. So very briefly, I will mention two areas in which I think the progress is really at high pace. One is genetics, and the second is what I would call the rebirth of tumor immunology. In genetics, all cancer is a genetic disease. All cancer are genetics. Most of them involve changes in DNA of a single cell that then expand to become a tumor. Lymphoma is also a genetic disease. But what is appearing in the last years is that the almost 80 different lymphoma types that appear in the WHO classification do in fact reflect a different genetic mechanism. Most of the work that is being presented here has involved still mutation in specific genes. So the coding genome has been dissected in the major lymphoma types, and there is an increasing number of genetic lesions that have a counterpart in a drug that has to be tested clinically for its activity against that lesion. One positive note that I think reflect a major effort is to simplify the enormous complexity of hundreds of genes that are mutated in consequences on a smaller number of pathways in the cells, and that makes diagnostic and therapeutics much simpler. One example is this drug that has caught the attention of hematologists lately, ibrutinib, that acts against the Bruton tyrosine kinase, a major transducer of the B-cell receptor, of the immunoglobulin receptor. And it's clear that while there is positive clinical results in CLL, in mantle cell lymphoma, for instance, genetics is helping a lot to understand where this drug should apply in the most common form of lymphoma, which is diffuse large B-cell lymphoma, by stratifying patients based on phenotype and genetics. So I think this, I expect will result in major savings and much more efficient clinical trials by addressing patient population that we know should respond as opposed to the one that should not respond. The second area that I think is the second aspect of genetics is important. It's important to note that the coding genome is 3 to 4% of the whole genome. Our DNA is making protein only in 3-4%. So far we have come in the major lymphoma or cancer type to exhaustion in the knowledge of 3 to 4% of our genome. The rest could be silent DNA, but also has an enormous role in regulatory functions that we know much less. There has been a few presentations that have addressed the fact that recurrent mutation occur in the non-coding genome of DNA, and we have to understand whether they involve microRNAs, long, short, non-coding RNAs, enhancer, super enhancer, major regulatory nodes that could have an impact again in the regulation of coding gene. This is enormous work that I think will develop in the next year, and there was some cutting-edge report here about the presence of recurring lymphoma type-specific mutation in the non-coding genome. Second area is what I call the rebirth of cancer immunotherapy. The mystery of why the immune system does not recognize transformed malignant cells has been hanging in the air for decades. And I think now we know, and based on a number of presentations here, that there are at least two mechanisms by which the tumors essentially becomes invisible to the immune system. Major development in the old cancer field, but now appearing very prominently here too, with major consequences that I think will be discussed in the next intervention. So there are, I think, schematically two examples. The tumor either genetically ablates molecules on the surface that are necessary for the immune system to recognize. I'm talking about major histocompatibility antigens that in a number of tumor types, including the most common one in lymphoma, they are genetically deleted, and therefore the cells become invisible to cytotoxic T cells and NK cells. And the second aspect is that there have been detected mechanisms by which normal cells are able to modulate the recognition by the immune system. And the tumors have been very intelligent in exploiting those mechanisms. I'm talking about the PD-1 and PD-L1 system, but that's only an example of more and more of this interaction between cells that by which cells modulate the ability of T cells to recognize. And tumors often overexpress this so-called immune checkpoint and therefore tell the T cells essentially do not intervention, although they recognize some non-self in the tumor cells. That has an enormous impact in the future, but I think has already showing very promising results in Hodgkin lymphoma and is being tested. There will be a number of presentations in other major types of lymphoma. Again, the challenge here, I think, for laboratory research, interfacing translational research, is to understand the responders from the non-responders. Could be genetically based and therefore more difficult to tackle the non-responders, but the recognition is important in order, again, to design clinical trials that address what we already know should be the responders. So that's, at this point, would be my overview.